Welcome to STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Cornegas, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's founder and Chief Executive Officer Emeritus, who is also the chairman of the Double Secret Selection Committee that selects all the guests who appear on STEM Talk.
Hi, Dawn. Great to be here.
So today we have Dr. Dominic D'Agostino, who over the past 10 years has been a frequent guest on STEM Talk. And today Dom joins us to provide an update on his recent research on ketone metabolic therapy, or KMT, ketone supplements, as well as hyperbaric oxygen therapy for traumatic brain injury. Dom and his lab at the University of South Florida have published more than 20 papers since Dom's last appearance on STEM Talk in 2023.
Dom is an associate professor in the Department of Molecular Pharmacology and Physiology at South Florida's Morsani College of Medicine. Dom has a background in neuroscience, molecular pharmacology, nutrition, and physiology.
And in addition to developing and testing metabolic-based therapies like ketone metabolic therapies, Dom's lab also investigates seizure disorders, brain cancer, neurodegenerative diseases, and rare genetic-metabolic disorders.
Before we jump into our conversation with Dom, we have a little bit of housekeeping. First, a big thank you to everyone who has subscribed to STEM Talk, and an extra thanks to all of you who have left those wonderful 5-star reviews. If you're new to the show and you like what you hear, please consider subscribing. And if you enjoyed today's episode, we'd love for you to take a moment to rate the show and maybe even leave a review. It really helps us reach more people who might enjoy STEM Talk.
As always, the Double Secret Selection Committee has been hard at work scouring iTunes and other podcast apps for the sharpest, funniest, and most lavishly praised field reviews to share on STEM Talk. If yours makes the cut and you hear it on the show, send us a note at stemtalk@ihmc.us to claim your very own official STEM Talk t-shirt, proof that the committee does, in fact, exist.
Today, our winning review comes from someone going by the moniker H. Coach Joe. The review is titled Science CPR. It reads, "This show really does explore the outer edges of science. They do so with such excellence that I wish I had more time to listen to each and every show. Dawn always seems to bring out the best stories of the wonderful scientists that she interviews. Ken always has great tie-in questions that make you think deeper about subjects you thought you had just learned so much about."
Well, thank you so much, H. Coach Joe, and thank you to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
Okay, and now on to today's interview with Dr. Dominic D'Agostino.
STEM Talk.
STEM Talk.
STEM Talk.
STEM Talk. STEM Talk.
Hi, welcome to STEM Talk. I'm your host, Dawn Cornegas, and joining us today again is Dom D'Agostino. Dom, welcome back to the podcast.
Great to be here, Dawn. Thanks for having me.
Thank you. And also joining us is Ken Ford.
Hello, Dawn, and hello, Dom.
Hey, Ken.
So, Dom, you've been pretty busy since you last appeared on STEM Talk back in 2023. Quite busy, actually. And you've authored or helped co-author more than 20 papers on a very wide range of topics over the past 3 years. You've also been an advisor to the University of South Florida trial on hyperbaric oxygen therapy for traumatic brain injury, and also been very active in what is known as the Metabolic Health Initiative, which is working to revolutionize medicine by advancing the importance of nutrition and metabolism in treating things like disease and improving people's health span, and also their resilience and performance. And so over the last 15 years, you've helped to advance the science and application of ketogenic metabolic therapy with your colleagues at Moffitt Cancer Center, with a particular focus on KMT to enhance immune-based therapies for several types of cancers. So this collaboration was some really good news for you, wasn't it?
Yeah, for sure. And it has been a growing collaboration. So historically, it started, back in 2008 with some interest in the hyperbaric of all things. And it gradually grew to where my students were, they would do their cancer research at Moffitt, and then we would write reviews with them. But they have become essentially a great transitional, partner or translational partner in this space for sure. So just moving the science into clinical application. So they do basic science, Moffitt does, but I just kind of view them as a transitional partner to move our basic science program into the clinic. And we've been hopefully successful in that with some latest grants.
That's really exciting work. And maybe ketogenic metabolic therapy, again, we call that KMT just for short, is a really good place for us to start our conversation with you today. And KMT is a dietary approach that focuses on a high-fat, low-carbohydrate diet to reduce glucose availability for cancer cells, potentially slowing their growth and improving treatment outcomes. And it's been explored as a complementary treatment for a variety of cancers, including gliomas, by shifting the metabolism of tumor cells away from glucose fermentation. So, Dom, it'd be great if you could give our listeners a sense of what is involved in KMT and just some more specifics around that research.
Yeah, so we had to come up with something other than the ketogenic diet to explain what we're doing. And ketone metabolic therapy, or KMT, is an umbrella term. I call it an umbrella term for a clinically supervised method to induce and sustain therapeutic ketosis. And that could actually be, as we've done work in the lab on time-restricted feeding, caloric restriction, ketogenic diets and different types of ketogenic diets, and also exogenous ketone supplements. And it is really like a personalization of the diet. For example, you take a patient and you personalize it to the foods that they will actually eat. And it's a personalization and also a precision approach to the diet. And precision approach meaning that there are defined biomarkers that define the diet. So KMT is defined by objective biomarkers, unlike any other diet. And I could go into that, as we discuss this. And this is really to enhance the safety and the efficacy of this approach and to make it clinically translatable. So KMT is an umbrella term for just therapeutic ketosis and a level of ketosis that's defined in the literature to have a therapeutic effect.
And just noting when we're talking about KMT, the suggested carbohydrate intake for KMT is less than 20 to 25 grams. And I'm sure most of our listeners are aware that ketosis itself is a metabolic state where the body shifts towards burning fat and ketones for energy instead of glucose. And that largely comes from dietary sugar and carbohydrates usually. Cancer cells typically consume glucose at a higher rate than normal cells, and this is due to a unique metabolic process where cancer cells convert glucose into lactate instead of fully breaking it down into energy. So this process really helps cancer cells grow and multiply rapidly. But cancer cells are pretty tricky, or maybe we can say devious, if we can say that, because they can adapt in unique ways, can't they, Dom?
Yeah, that's the problem, targeting cancer and different types of cancer too. So that, every cancer sort of has different molecular pathways that can activate, for example, angiogenesis. And, when tumor is deprived of oxygen or nutrients, it grows blood vessels faster. For metabolic reprogramming, to basically, enhance the tumor's metabolic vitality through autophagy, through something called glutaminolysis, which is the ability to use amino acids like glutamine for fuel. So we never assume that ketone metabolic therapy alone is sufficient to stop cancer growth, but it sets the stage that typically slows cancer, and it primes the tumor microenvironment to sensitize it to other types of therapies. And this was kind of the rationale for the review article that we wrote with the Moffitt Cancer Center entitled Targeting the Warburg Effect for Glioma. And so there, it's multi-targeted. Our approach is multi-targeted. And ketone metabolic therapy is really an adjuvant and a supportive therapy for other modalities to enhance the efficacy of other modalities. And that's really important because some people will make the claim that ketogenic diet can cure cancer. And when they make that claim that I see online, I cringe. And that is not something that we promote in our in our lab or in the clinic. But it does. It is a supportive adjuvant approach for other forms of therapy.
That's a great clarification, Dom. Thank you.
KMT has particularly shown promise in treating high-grade gliomas, which are a type of brain cancer. Glioblastoma, or GBM, is the most aggressive primary brain tumor in adults, and it typically carries a relatively lethal prognosis. You are part of a massive review titled Clinical Research Framework Proposal for Ketogenic Metabolic Therapy in Glioblastoma. In this review, the team that was put together for this paper proposed guidelines for the management of glioblastoma based on an understanding of cancer as a metabolic disease, in particular involving mitochondria. Can you give us an overview of this review and its key takeaways?
Sure. Yeah. Thanks for bringing up that review. That took a couple years, quite a few years to get that out. The ketone metabolic therapy framework paper was really focused on standardizing the clinical research for ketone metabolic therapy for this most aggressive form of cancer, glioblastoma, with a high emphasis on, enhancing the safety, the efficacy, and also the clinical translatability of this approach. In particular, targeting therapeutic ketosis with something called the glucose ketone index, achieving and maintaining that, designing a strategy to achieve and maintain low glucose, high ketones, and then to link that biomarker to the outcomes. And that could be a CT PET scan, that could be CA19-9, a common cancer marker, or even PSA, prostate specific antigen. So having, a very defined clinical metabolic biomarker and then correlating that to outcomes is going to be very important. Also, the review was written to really emphasize personalizing this approach and of course, adding other forms of metabolic therapy. And that includes metabolic drugs, metformin, different glycolytic drugs, and also setting the stage for oxidative stress therapies, which could be the standard of care, chemotherapy, radiation, or even immune-based therapies.
Really interesting and very hopeful. Additionally, from a patient advocacy perspective, this review also recommends that there should be an aggressive education campaign that can arm patients with knowledge about KMT, as well as other novel therapies. And I guess we're playing a role in that today, but can you talk about that recommendation some?
Yeah, education outreach is really essential, particularly in this topic. It's the type of research that people see and they try to implement themselves. And then they also, they take certain aspects of the research and they can promote misinformation, which kind of comes back on us in a negative way. I feel like I'm always fielding that. But we're trying to prevent misinformation with putting this out there, trying to prevent extreme diets, untested interventions like juicing. Probably the biggest category of dietary therapies for cancer is juicing, right? So that's not a really good approach. Or even unsupervised use of different drugs that are gaining popularity, like many drugs out, like ivermectin, for example, which actually can target things like cancer stem cells and mTOR and decreased metastasis, in animal models, but, preventing the unsupervised use of these things and also just highlighting the emerging research and defining very specifically the toxicity of these potential compounds or approaches, and also defining what the research says and sort of outlining safe ways to use these approaches with the emphasis on clinical trials. But we kind of leave it at that. We put in a lot of supplementary information in this review. There are I think a half dozen supplementary files associated with that review, and it really goes through all the different types of drugs that could be used in synergy with ketone metabolic therapy.
Absolutely. I think the educational component is so necessary, in particular, as you said, with respect to alternative treatments and things like KMT. As I mentioned earlier, this is a really massive review that started out at something like 200 pages and, of course, had to be cut down for publication to around 50 pages. With 49 authors, this had to be a tough process. I can't imagine cutting from 200 to 50 pages with 49 stakeholders. That had to be interesting.
Yeah, it was very interesting. And, everybody has different opinions. And I think the ultimate, paper that came out of this is a very refined approach to this. And there are people, it was a team effort for sure. Like Dr. Thomas Seyfried is the senior author, and Dr. Tomas Duroc, who's the first author, did a lot of heavy lifting. But also people that Ken, like Adrian Scheck, Colin Champ, Pavel Klein. There's neuro-oncologists. It's really important to have the people sort of in the trenches with the patients. There's neuro-oncologists like Jethro Hugh. He's an NIH-funded investigator, like, essentially using this platform, using this review article in a current clinical trial. And also he's at UCLA, and Chris Smith from the Barrow Neurological Institute. He's a brain surgeon, also using this approach. So we had basic scientists, neuro-oncologists and clinicians. And it really helped to define, to cut this down to 50 pages. And what we couldn't include, we included in the supplementary information that you can also download.
Good. I was hoping you would say that.
Yep.
We'll be having a link to this paper in the show notes for folks who are interested.
So, Dom, back in 2024, you were part of another paper, and it was titled Targeting the Mitochondrial Stem Cell Connection in Cancer Treatment, a Hybrid Orthomolecular Protocol. So this paper looked at the mitochondrial stem cell connection theory, also known as MSCC, which suggests that cancer originates from chronic oxidative phosphorylation insufficiency in stem cells. So this insufficiency leads to the formation of cancer stem cells and abnormal energy metabolism, ultimately resulting in malignancy. There were 16 research centers and other organizations involved in this paper, which introduced a hybrid orthomolecular protocol targeting the mitochondrial stem cell connection. And this connection is a relatively new concept, which combines cancer stem cell theory with the metabolic theory. So first of all, it'd be great if you could give us an overview of the mitochondrial stem cell connection. That would be a great place to start with all this.
Yeah, I'd like to just credit Otto Warburg because, it's this also results it stems off of the Warburg effect, which is essentially says that damaged mitochondrial respiration leads to compensatory fermentation. That's why cancer cells are very glycolytic, and that's why they use, upwards of 100 to 200 times more glycolytic activity than healthy tissue surrounding it. So mitochondrial dysfunction creates this Warburg effect, and that basically decreases the bioenergetic effect of the cells themselves. You have damaged the mitochondria so that the ATP levels fall and the cells sense energetic crisis, and that kicks on oncogenes, which then endows the expanding biomass with more ability to ferment and grow. And out of that fermenting, growing tumor are these cancer stem cells. And I think of cancer stem cells as like a queen bee. A queen bee can kind of give rise to an entire colony, right? So these cancer stem cells are very prolific, and you have cancer stem cells that are highly glycolytic, highly prolific, and within the mix, you also have these tumor-associated macrophages. And the cancer stem cells and the tumor-associated macrophages kind of grow together, and that's what's really responsible for a consolidated tumor, for example, in your liver, to be invasive and metastatic. Most people don't die of a tumor per se, unless it's in the brain, but still you have invasion, in the brain. They die of the metastatic process. To summarize, cancer stem cells, they are highly glycolytic, and they also endowed with the ability to use amino acids like glutamine. They can also use fatty acids, not so much for fuel, some of it for fuel, but also mostly for biosynthesis of the expanding tumor biomass. And cancer stem cells can also use more cholesterol. So you can use a drug that's a HMG-CoA reductase inhibitor, like a statin even, or antibiotics that decrease protein synthesis. And these are all approaches that we've incorporated into the paper. Like a stem cell, cancer stem cell is just a very aggressive cancer cell that's like a queen bee and can give rise to a massive colony of tumor cells. And they have very unique abilities that this paper aims to target.
So, Dom, in this paper, you and your colleagues propose a protocol that would enhance oxidative phosphorylation and also inhibit the primary fuels of cancer, glucose and glutamine. So that would target both the cancer stem cells, queen bees, and metastasis. And this is just referring to how cancer cells spread from an original tumor to other parts of the body. So can you explain why this concept is attracting so much interest as a potential therapeutic approach for cancer?
Yeah, well, I think this approach is really targeting the heart of the tumor growth and proliferation, which is the stem cells. And I like to emphasize it is a theoretical approach. And it's based on emerging evidence-based science. And I'd sort of like to emphasize that. So basic science research, case reports, and there's many authors on this paper, right? And they have a lot of experience implementing various aspects of this and sometimes together as a whole on many different patients. So it evolved from that. But an orthomolecular approach is really kind of the definition is it's targeting kind of metabolic deficiencies. And an example of that would be we study a wide variety of different disease processes, including ALS. The Diana protocol is something that we've published a couple papers on. And in ALS, alpha-ketoglutarate and GABA are therapeutic because of various deficiencies, and that could be therapeutic. The same thing kind of would apply from a cancer perspective. And there are many different ways to do nutritional and metabolomic profiling to understand a person's metabolism and then to adjust the diet based on metabolic biomarkers and also potential, I guess what I call metabolic bottlenecks. And when you address some of those issues with an orthomolecular approach, the therapies tend to work a lot better. And then it opens up the potential to use various drugs that are talked about, in the protocol that can enhance the metabolic effects. So if we can change a person's metabolism to metabolically target and minimize tumor growth, and then that sets a stage for a number of other drugs in the protocol that can further target very specifically the cancer stem cells.
So Dom, this is a great overview. So can you start with the orthomolecular protocol, which is an approach that focuses on preventing and treating diseases by correcting nutritional balances in the body? Is that correct? And you kind of addressed this, but can you dig into that a little bit more of what that looks like?
Sure. Yeah. And, in the world of functional medicine, a number of these, metabolic tests and functional testing is kind of standard, but in the world of conventional medicine and even oncology, they're a bit fringe, although they're becoming more mainstream. And, I like to use various clinically validated or clinically used metabolomic testing. And there are a number of different companies out there that offer these tests. I'm not going to name the companies, but you can look them up. That would basically, you do a combination of organic acid, blood testing. You could do a cheek swab to get genetics too. And then that information is analyzed by the lab and you get a report back. And something like organic acid testing or metabolomic testing is actually the standard of care for inborn errors of metabolism and also in newborn screening. So these approaches that I'm talking about are actually routine and the standard of care for different types of disorders historically for decades now. So what's different here is that you're applying this validated methodology to this particular scenario, so the orthomolecular approach to cancer. And you can find very specifically various deficiencies and metabolic bottlenecks and correct those in the patient. And then what you're doing is you're setting the patient up for a successful, so for example, outcomes are significantly better if you improve some of these nutritional deficiencies. Like no one would argue that omega-3 fatty acid status is tightly correlated with cancer outcomes, or even B12 status, which is deficient in pretty much most people that are old or on a wide variety of different drugs. And then you apply, if you correct the metabolism, augment the metabolism, and then there are a number of different drugs that I won't get into now. I just encourage people to download the paper because we actually list the drugs and we also define the dosages of the drugs. And keep in mind that this is completely, theoretical, but it's being used in various clinics. And these drugs further target the molecular stem cell processes. And for example, like metformin is something that we all know. One of my students, PhD students, did his PhD dissertation on metformin and DCA, which are mentioned in that. So metformin is a mitochondrial toxin, believe it or not. That's why you don't want to use it if you're like an athlete, right? But in the context of cancer, it can be very efficacious by increasing reactive oxygen species. It activates AMP kinase. So AMP kinase activation has a pretty strong anti-cancer effects. It also inhibits mTOR. So, many cancer drugs are trying to target mTOR. And so metformin is kind of one, it's in over 200 clinical trials. So that's part of it, a well-known drug. And there's also drugs in the protocol that inhibit, for example, glutamine or glutaminolysis. So like sodium phenylbutyrate, right? And it's used for urea cycle disorders. So that can lower blood levels of glutamine and glutamine availability to the tumor. The two primary fuels that cancer stem cell will use are glucose and glutamine. So that's, these are ways to inhibit that. And and these drugs by themselves are typically not very efficacious as a standalone, but when they're combined with a metabolic diet or a dietary approach, then they become quite synergistic.
And speaking of the dietary approach and the dietary intervention involved in this, is there anything particular about that apart from, the typical sort of ketogenic diet?
In the orthomolecular approach?
Yes.
Yeah. So that's a good question. So with the orthomolecular approach, it's really about adjusting and titrating the diet to personalize it to the patient with precision types of tests that can be done. But you can achieve and maintain therapeutic ketosis with a ketogenic diet, time-restricted feeding, caloric restriction. And these things are not mutually exclusive. For example, like you can do time-restricted feeding with the ketogenic diet and also do a form of caloric restriction. But in addition to that, unlike what mainstream oncology it kind of does not do, you want to look at just basic things like your vitamin D3 levels, because that's going to influence outcome. So this is something that's not standard, but should be. Like I mentioned, B12. As we age, our intrinsic factor, which allows us to absorb B12, goes down profoundly. So we're only, absorbing a fraction. And also drugs in the protocol, like metformin, or even common drugs like proton pump inhibitors or GLP-1s. These drugs are very well known to lower B12 levels. And B12, like if your B12 is deficient and you undergo chemotherapy, you are much, much more likely to get something like peripheral neuropathy or even atrophy of the brain. So just like simple things that is in the protocol that really should be the standard of care. And I think science is moving in that way. But to look for things like, to supplement things like selenium, alpha lipoic acid, and acetylcysteine, and omega-3s, or just, check your omega-3s so you're not deficient, because omega-3s are tightly correlated with prevention of cancer, but also improved survival if you're going through. So that would sort of encompass aspects of the orthomolecular approach in regard to diet.
That's a good explanation. Let's switch gears here and talk a little bit about ketone supplements. You recently published a paper titled Divergent Hepatic Outcomes of Chronic Ketone Supplementation. Ketone salts preserve liver health, while ketone esters and precursors drive inflammation and steatosis. There is a lot of interest in exogenous ketone supplements because they substantially elevate circulating ketones without carbohydrate restriction, or at least without as much. The problem is the long-term hepatic safety of ketone supplements remains a little bit unclear. In this rodent study, you evaluated the formulation-dependent impact of chronic ketone supplementation on liver histopathology, inflammatory signaling, and systemic biomarkers. First of all, can you give us a little bit of an overview of the currently available ketone supplements, breaking them up into their different categories and their various formulations, as well as their potential applications, and perhaps most importantly today, any cautionary considerations?
Yeah, sure. Glad to do that. It's, an emerging area of high interest, and and a lot of people look at this very, critically, as they should, as they did with something like creatine monohydrate in 1992. When it came, it took about at least a decade for the science to sort of really validate the early studies. And there is a paucity of studies, of chronic studies with ketone supplementation. And I think that's kind of what our paper addresses, really just kind of, highlighting what can happen if you consume these ketone supplements, or certain ones, chronically over a period of time, and also age is a factor too. So the different ketone supplements include beta-hydroxybutyric acid, which comes usually in a very acidic solution, and then you dilute that and you can drink. That was like the first ketone, and then that's something that we first started playing around with. And then the ketone salts, and that could be just simple sodium beta-hydroxybutyrate. So beta-hydroxybutyrate can be combined with different minerals ionically. So that's sodium, potassium, calcium, magnesium, and that creates a ketone salt. And they could be formulated in a way to be an electrolyte supplement, which are all the rage now, right? So you can have an electrolyte supplement, and the electrolytes deliver the electrolytes, but also the ketones. So the ketone salts. And then you have another type of ketone that's not really a ketone, but it's a precursor. It's a 1,3-butanediol, and that's a diol. And if you consume 1,3-butanediol, the liver sees that as an alcohol. It's an alcohol dimer, or also called a dialcohol, and it goes through that same metabolic pathway that ethanol goes through. So, ethanol creates acetate, and 1,3-butanediol goes through that pathway and creates beta-hydroxybutyrate. And interestingly, acetate from ethanol is a very good alternative fuel for the brain. And that's another area of interest that I have looking at acetate. And then 1,3-butanediol is often used to make what's called ketone esters. So you can take 1,3-butanediol and put 1 or 2 ketone molecules, beta-hydroxybutyrate or acetoacetate. The issue is something like a ketone monoester is more than 50% 1,3-butanediol, and that could be potentially problematic. The diester is about 30%. And then there are ketone supplements like MCT, medium-chain triglyceride, that we know. And we're working on a variety of other ketogenic molecules that are ketone esters from glycerol, or the ketones that have a sugar backbone. And these have very interesting properties too. But I will say that any formula, like ketones, exogenous ketone in and of itself as a standalone therapy is less efficacious than when these things are formulated together. And I think that's an important thing. We always see benefits if you formulate different compounds together.
So, Dom, it's really interesting. So in this study, you and your colleagues evaluated a range of blood chemistry markers in rats that were chronically fed either a standard diet or a standard diet supplemented with low-dose ketone ester or a high-dose ketone ester. And the goal of that study was to determine whether ketone supplementation induces significant changes in liver and kidney biomarkers, lipid metabolism, or even blood biochemistry. So what did you all find in the study?
Yeah, this study kind of started a long time ago, well over a decade ago, and it was part of an Office of Navy Research grant. We were studying at the time, as you know, Don, we were looking at the ketone ester for central nervous system oxygen toxicity. And in the context of that use, you're only giving a dose of ketone ester acutely, and then 30 to 60 minutes later, you're looking at the effects of reducing seizures. In parallel to that, we did studies where we gavage, where we tube-fed ketone esters and different doses of ketone esters and 1,3-butanediol over a period of time. So that was 28 days, but also 100 days, also incorporating the exogenous ketones into the diet itself or the food itself. And what we found was a little bit alarming in regard to the blood work was not significantly altered all that much. But when we sacrificed the animals and did the histology, we saw some potentially, alarming things in the histology. So changes in the histological features of the liver. And that included something called sinusoidal dilation, where there was we saw fatty liver, and we saw expansion and kind of holes in the liver where fatty acids were being accumulated. We saw signs of the liver that were essentially pre-necrosis. So we saw changes in the histology that typically precede necrosis or apoptosis. And we saw some changes in the lipid markers and reactive oxygen species and inflammation. And that was sort of the take-home message, is that when these compounds are consumed over a protracted period, the liver does have to work considerably harder. And we did not see major functions in the AST and ALT, the liver function tests, but that's kind of common too with non-alcoholic fatty liver disease. So, which is ubiquitous especially in the United States, is that the damage is already done before the liver function tests really show it. And typically you see it in ALT, and then AST will start climbing up. But if you do a CT scan or even a high resolution ultrasound, you already have the fatty liver and the damage is done. So the histology is kind of the canary in the coal mine, and is telling us that these ketogenic compounds, or certain ones, are not inert when it comes to liver function.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
How confident are you in the rat model, which this was done in, translating to humans? You know, you see a lot of good translation, but you also see a lot of times it doesn't, particularly in things like drug-induced or alcoholic liver disease. And what do you see as the next step?
Yeah, that's a good question. I think animal data is informative, but not always translational. But it's also kind of important to acknowledge that 1,3-butanediol is an alcohol. So it goes through that alcohol dehydrogenase, aldehyde dehydrogenase pathway. So we know, basic biochemistry says that. And I think what hopefully our study will do is there's a paucity of chronic studies in humans looking at these compounds, 1,3-butanediol and 1,3-butanediol associated esters for long-term chronic use. I mean, meaning like, 12 weeks, 16 weeks, or even a year. I think there's 2 studies and one's a case report. So there's not a whole lot of studies that really inspire confidence that these compounds consumed in a prolonged period of time would be safe. So I think that's really the take-home message, that there's a precedent here that we need to be aware that particular ketogenic agents that are formulated with 1,3-butanediol have the potential to be, in addition to the potential liver toxicity, is that it's well defined in the literature and animal literature that 1,3-butanediol causes withdrawal and dependence, meaning that if you feed 1,3-butanediol and then you take it out of the animal's diet, the animal goes through withdrawal symptoms similar to ethanol, and there's also dependence created with 1,3-butanediol. And from a clinical standpoint, 1,3-butanediol in a recent paper was given at the maximum dose, which is 34.5 grams, and 40% of the participants, which were young, experienced dizziness. So that's kind of an alarming thing that was kind of buried in the paper. My concern is that older people that are trying to target cognitive function and dementia and Alzheimer's disease are already at sort of a metabolically and neurologically very vulnerable to getting dizzy. And since 40% of younger people experience dizziness with 30 grams per day, which was even divided, that 34 grams was divided into 2 doses to make the subjects less dizzy. So my concern is that not only the toxicity, but since 1,3-butanediol is a narcotic dialcohol, that older people consuming chronically large doses of it could be potentially getting dizzy. And I know just from consuming this myself that you do get a buzz from it. And I think it could be a good alternative to alcohol if someone's trying to kind of get off of alcohol. But these are studies that really need to be done. And I think this idea behind doing this, this paper was to highlight the need for more long-term studies.
Back to the paper for a minute. Could you talk about how the dose, the high dose and the low dose in the rodent model relate to typical human doses?
Yeah, I think that's an important concept. So for the higher dose, which was most problematic, that would correlate to someone like me consuming about 1 gram per kilogram per day. So that would be like me consuming 30 grams, 3 times a day with meals. And
yeah, that's a huge dose.
That said, that's a large dose, but 30 grams of 1,3-butanediol consumed 3 times per day is actually what's needed in me to keep my levels of ketones at 2 millimolar throughout the duration of the day. So, because 1,3-butanediol causes a slight rise, but if it was maybe the ketone ester, that would be kind of higher, but a ketone ester is also more than 50% 1,3-butanediol, or at least a ketone monoester. So if I consume, and other people have sent me blood work, consuming 1 gram per kilogram per day for more than 2 weeks, you're going to see a bump up in your liver function tests, including AST, ALT, and also GGT. GGT will be the more sensitive indicator of alcohol-induced liver stress, kind of like the canary in the coal mine. And you can also look at the size of the red blood cells. So, and I would encourage people who are experimenting with these drugs because, an 80-year-old has about an 80% less ability to detoxify alcohol. So the older you are, the more of this could be a concern, especially if you're using these things chronically. I would encourage people to do a comprehensive metabolic panel to look at their liver function test before and after if they're using 1,3-butanediol or 1,3-butanediol associated esters. And taking a step back, this was part of an O&R project, and it was really not too much of a concern for us because the primary application of a ketone ester for oxygen toxicity seizures is to deliver an acute dose, like prior to diving. It wasn't to consume these things chronically. So there's less of a concern there. But, that was well over a decade ago. And then the whole market of ketogenic supplements kind of exploded after that. And then you have people chugging these things every day. And some of them are older and they want to maintain those ketone levels high throughout the day. So we thought it was probably important to get this information out there because no one had done these types of studies.
Absolutely. And in February, Ben Bickman's lab published a paper which you helped co-author that reported on a study which examined the effects of ketone supplements on liver function, just as we're discussing. And Ben was our guest on episode 143 of STEM Talk. Can you talk about Ben's study a little bit and its findings and how it relates to the study we just discussed?
Yeah, Ben reached out to me when he started the study and he had some questions about dosing. And he actually used lower doses than I used in my study, and he only did it over a week's time. And what was really interesting about Ben's study is that it validated the work that we did in our lab, in an independent lab, and he used mice and we use rats. Essentially, what he found is that a 1-week administration, which is not chronic, it'd be like subchronic for sure, he saw some very significant findings in regard to the looking specifically at the liver cells. And he showed very convincingly that administration of 1,3-butanediol, unlike just giving beta-hydroxybutyrate in the pure form, the D enantiomer and the L enantiomer. So he was very innovative in looking at those two enantiomers, which generally had positive effects, like no effect or positive effects. But unlike pure beta-hydroxybutyrate, 1,3-butanediol caused a remarkable depletion in ATP levels and also elevated inflammatory markers and elevated reactive oxygen species or free radicals. And this sort of, he has a wide variety of tools to study this, including the Ouroboros system. And I had an opportunity to deliver a talk at Brigham Young University and see his lab and kind of envious of some of the equipment and that he has to run these studies looking at the metabolic effects of these things. But it kind of validated our work showing the potential toxicity of 1,3-butanediol. In particular, it showed very convincingly that 1,3-butanediol depletes liver ATP and elevates inflammatory markers and elevates reactive oxygen species. Keep in mind that this is in the liver. And also keep in mind that the liver has to do the work of converting the 1,3-butanediol to the ketone bodies, and the ketone bodies are beneficial. So the 1,3-butanediol generated ketone bodies could potentially be negating some of the effects of the 1,3-butanediol, if that makes sense. And I'd also like to emphasize that when we drink alcohol and we're addicted to alcohol, the ethanol that we're consuming is converted to acetate. So acetate levels are elevated in the blood, and then acetate becomes one of the primary fuels for an alcoholic's brain. And when you get off of ethanol, you go through withdrawal, and that's due to acetate withdrawal, believe it or not. So that's an interesting area of research. And I was involved in a PhD dissertation that was looking at acetate as, an alternative fuel, but also to supplement ketones, and one of them, 1,3-butanediol, or the ester, as a less toxic way to wean someone off alcohol or ethanol. And there are clinical trials right now on clinicaltrials.gov that are exploring this hypothesis that was tested in rats, but in humans. So, but I think that we are pivoting away from 1,3-butanediol and 1,3-butanediol associated esters because there are safer and more potent ketogenic compounds that can be used for the various applications. But I think with that said, there's already, good data with 1,3-butanediol associated ketone esters, which we have published, showing that these things can be remarkably beneficial and therapeutic, but it's very context specific. And we do lack, the long-term studies, especially in humans. So just emphasizing that, these studies need to be done, and hopefully they will be done. And I'm also very excited and encouraged about the new ketogenic compounds that are in the pipeline.
So Dom, just another little bit of a pivot, just great work, by the way. So last year, our friend and colleague Andrew Kutnick joined us on STEM Talk episode 185. And you recently worked with Andrew on a paper about carbohydrates and physical performance, which has become quite popular. A lot of discussion around it. So the paper is titled Carbohydrate Ingestion on Exercise Metabolism and Physical Performance. And this appeared in Endocrine Reviews, and this paper showed that a small amount of carbohydrates is all you need for athletic performance, and going above that doesn't really provide any additional benefit. So this is an interesting article because carbohydrate ingestion before and during exercise has long been seen as necessary for peak performance, and definitely can attest to that from the races that I do. It's all about carb loading and maintaining carbs, right? So can you please discuss this paper and its findings and implications?
Yeah, this was a massive paper, and big props to Andrew Kutnick and Tim Noakes for doing like a lot of the heavy lifting, on this paper. And it was published in Endocrine Reviews, quite a high impact journal. And we were notified last week that this was the highest impact review article, I think, in there. So it's like they reached out to us. And Andrew did like a lot of the heavy lifting on analyzing the data, but the findings were very, contrasted what we are taught and contrasted even what's in the literature, because no one has really analyzed it from this perspective. And the take-home message, I think, for the listener out there is that carbohydrate supplements and sugar supplements are definitely overhyped and oversold and over-commercialized, I guess you will, for the athlete. And an analysis of the data shows that during exercise, far less carbohydrate is needed, 10 grams per hour to 20 grams per hour for a larger individual is needed. And this is much, much lower than the general recommendations. And keeping your body somewhat hungry for glucose allows your body to be more metabolically flexible. So you're pulling off of so you can maintain metabolic flexibility and tap into your fat reserves more continuously. It is very context dependent. It's dependent upon how the athlete has trained. So if your body is fat adapted or ketogenically adapted, the size of the individual, and also your GI tolerance to sugars is also a big thing too. So if I could not personally down, 100 grams of sugar per hour and just would not settle well. But I think the important take-home message is to titrate the amount of carbohydrate starting low and titrate it in a way that avoids excess energy. We call that energy toxicity, which of sugar, and which will cause excess glycolysis. And that essentially is training your metabolism to rely more on glucose and glycolysis, and that could train your metabolism in a way that could deplete your liver glycogen stores faster, and you can actually bonk faster. So I think the thing is to think about your fuel is kind of like a rheostat, and you can adjust your carbohydrate, keeping it as low as possible, but adding a little bit more per hour for the athlete, is because there's no additional benefit to these super high levels of sugar that are often advocated, and they could actually do more harm than good, not only to your performance, but in previous research that we published showing that adherence to what the athletes are promoting, these high carbohydrate diets, show that wearing a continuous glucose monitor, there was subjects in the study that we published about 3 years ago showing that they're essentially pre-diabetic. And especially if you look at their CGMs at nighttime, are hovering above like 100 during the night, and they are essentially pre-diabetic. And then when those same athletes switch from high carb to a carbohydrate restricted ketogenic diet, that normalizes their circadian glycemia. And an important message is that if they were to follow that high carb diet, and not, or even exercise or not exercise, that's going to promote a trajectory to metabolic dysregulation and diabetes. And that's essentially what happened to Tim Noakes, right? So I think that motivated him to investigate this topic of being more fat adapted and metabolically flexible. And that can pay big dividends, not only on performance, in particular endurance performance, but also in metabolic health for the athlete.
Yes, I see. I have, at my age, I've got friends that are age group athletes in endurance type sports. Nothing I'd be interested in, but they seem to enjoy it. And several are pre-diabetic, and they're thin, and maybe it's partially not enough muscle for glycogen store, but I think it has a lot to do with their diet and with their reliance on carb loading. But I could be wrong, but it sounds like you've seen that with people wearing continuous glucose monitors.
Yeah. Well, we published on that, and about 1/3 of the participants in a study that we published prior, Andrew, I think, was the first author on that too, showed that the high carb diet really made them pre-diabetic, and that would be flagged in their blood work. And looking at metabolomics flux data with some of even the data from my own self, my own data gets flagged on the platform as it gives a red flag for being glycolytically restricted. And it says that my physiology has a bottleneck in glycolysis. So that gets red flagged on the AI analysis of my metabolomics data. And it shows my lactate is too low, my urine succinate is too low. It shows 3 or 4 bottlenecks that shows I have a restriction in glycolytic flux, but then it shows my fatty acid and ketone flux is pinned, and it's like off the charts. So in my view is that that's a good thing, and that the actual analysis is based on high carb adherence. And I think it's really beneficial for the athlete to be metabolically flexible and keep their metabolism hungry for glucose, and that will help push them to more fatty acid and ketone metabolism. And just an overconsumption of sugar can lead to this energy toxicity and excess glycolysis that if you run out, you rapidly deplete your liver glycogen. So ketones and fats are glycogen sparing. This is a concept that Jeff Volek and Stephen Finney have been advocating for over 2 decades before I even got into this research. And it made a lot of sense back then. And I think now, or Jeff Volek was also, just like to highlight, he was an author on this paper, and it's kind of validating what they've been saying for 2 decades now.
At the beginning of our conversation today, we mentioned that you have been an advisor to a University of South Florida trial on hyperbaric oxygen therapy for traumatic brain injury. TBI is something really interesting and important here as well. How is that trial coming along?
Yeah, thanks for asking. That particular trial, the use of hyperbaric oxygen for traumatic brain injury in veterans with and without PTSD, is coming along great. And we meet every 2 weeks kind of as a team and just talk about how it's going. It is blinded, so I don't have any. I think the first data set will be unblinded soon to look at some of the outcome measures. But I'm involved in various aspects of biomarker monitoring and mechanistically, things to look at in post-analysis. What's really interesting and different about this particular hyperbaric oxygen study is that the sham, or the control, is a proper control. In previous studies, the control was hyperbaric air for some of them, but the Israeli groups, I think, were first to spearhead like a control where you can pulse the pressure in the beginning to make the person in the chamber think that they're undergoing hyperbaric oxygen, but they stay at 1 atmosphere. And then we have basically like psychologists that will interview the subjects, and it has been confirmed that they have no idea. I think they actually trend towards like the opposite of feeling that the control was the hyperbaric. So essentially, they cannot detect if they're going through hyperbaric, and that's a unique aspect. But this is by far the biggest and largest, most comprehensively designed hyperbaric oxygen study for TBI, and it's a $28 million funded project. So, and my understanding is that if there is a benefit to hyperbaric oxygen therapy for traumatic brain injury, that the people in the trial will have free access for at least a year or so to the hyperbaric after the study. But it involves brain imaging and the assessment of every biomarker you could think about for this particular study, because the funding level is there to do that. So I'm kind of waiting very enthusiastically to see the data as it emerges. And we're about, I think, a little under, about halfway through at this point in time. And in addition to that, we're setting up to hopefully do some preclinical animal model work to look at diffuse axonal injury, which is a more severe form of traumatic brain injury, and to really look mechanistically of what's happening there and how hyperbaric oxygen may play a role in mitigating that, in addition to various metabolic therapies that we're talking about here, and working on an NIH proposal to hopefully in parallel. And the protocol will be very similar to the hyperbaric protocol will be similar to what's being done here, but in a preclinical model.
Dom, that's really exciting and something that's definitely been needed in the hyperbarics community. So kudos to you all for conducting that study. And you've become quite well known for your hyperbaric associated research projects. And that's even how, we met back in the day. And you recently had an editorial in Frontiers that gave an overview of the emerging applications for hyperbaric therapy and hyperbaric oxygen therapy in the treatment of different neurological disorders. So what were some of the key points in that editorial? Can you just give us a brief overview?
Yeah, sort of at a high level, some of the take-homes, I guess, would be that hyperbaric oxygen is a dose-dependent therapy, and it's not, not a single intervention. And the clinical outcomes really depend on a combination of pressure and oxygen, and these things can be working independently. So that's sort of, and we need more clarity and more. Hyperbaric oxygen is kind of like the protocol is just a blanket protocol. Right. And I think we need to understand mechanistically how we can titrate that to the patient. And different neurological outcomes require different hyperbaric oxygen dosing strategies, right? And I think that was a big one going into this TBI trial too. And I think the Israelis do the air breaks, during the hyperbaric to prevent CNS oxygen toxicity. And that's kind of a little bit slower to adopt here. But in the context of things like stroke or neurodegeneration or traumatic brain injury, obviously, is that the potential for CNS oxygen toxicity is quite higher. So I think that it's not a one-size-fits-all approach, and we have to consider lower dosing protocols and titrating them up to meet the therapeutic needs, and also with the air breaks. And both hypoxia and hyperoxia drive biological effects. And the air breaks that were done by the Israelis, and they've also spearheaded, looking at hypoxia and coupling it with hyperoxia for activating, HIF-1 alpha and stimulating erythropoiesis to boost your red blood cells. This is something that and athletes do things like that. Those relative changes from hypoxia to hyperoxia, or even normoxia to hyperoxia and pulsing, your body senses that relative change, and it's kind of sensing it in a way that it's sensing hypoxia. Do you know what I mean? From dynamically changing the interplay of oxygen delivery. And even though you're not hypoxic, your body is sort of sensing it as relative hypoxic signaling. And that affects gene expression pathways, mitochondrial function, and neuroplasticity mechanisms. So I'm talking with a group now, and they really feel that that's very important. The neuroplasticity mechanisms associated with these protocols are having profound effects on post-traumatic stress disorder. And they've already published a number of randomized controlled trials to show the benefits of hyperbaric oxygen using this protocol for PTSD. And I think, as you know, Dawn, we wrote that review together. Hyperbaric oxygen induces pleiotropic mechanisms beyond oxygenation, reducing inflammation, enhancing mitochondrial efficiency, angiogenesis, neurogenesis, improved blood flow, and then, potentially increasing stem cell production and releasing those stem cells into circulation, and they can hone in on different, sites of injury. And we know that happens in the context of wound healing, but the brain, traumatic brain injury, is also a wound, right? And we think that those mechanisms may also be at play with traumatic brain injury. But I think we really need precise dosing is critical for efficacy and for safety. And the field really needs standardized evidence-based dosing frameworks, similar to the ketone metabolic therapy framework for GBM. We need like a hyperbaric, evidence-based framework for, the use of hyperbaric for particular disorders.
I couldn't agree more. And again, really excited to see that your group has been working on developing that, which is really exciting for the field. And just thinking too, just to note, there's a lot of excitement around different applications for treatment, but also from the neuroprotective side of things. So a lot of different areas that we could really start to tap into once this dosing piece gets worked out and we actually can run these trials in a meaningful way. So really excited to keep following this work, Dom.
Thank you.
So going back to the TBI discussion, you recently gave an evening lecture at IHMC on TBI and populations that are most at risk for traumatic brain injury. So for listeners who are interested, a video of Dom's lecture is available on IHMC's YouTube channel. For brief background, though, Dom, can you give us a description or explanation of how an injury to the brain can result in neurometabolic crisis?
Yeah, sure. Traumatic brain injury is a growing concern, even a crisis, especially in older adults. And traumatic brain injury initiates a neurometabolic crisis. And you have an impact, the injury itself, and then that quickly causes a neurometabolic crisis. The first, actually, few hours, you get a hyperglycolytic effect. And then you get this protracted glucose hypometabolism in the brain. That is a hallmark characteristic of chronic traumatic encephalopathy, for example, and that glucose hypometabolism leads to things like post-traumatic epilepsy, depression, and psychiatric disorders. So you have the impact and the energetic crisis, which is the driver for the neuroinflammation and not the opposite way around. And now new evidence, now this is becoming more accepted. So you have the energy crisis, impaired energy from blood flow, for one thing, low levels of oxygenation, and just impaired glucose metabolism. And that neuroinflammation then further exacerbates the energetic crisis. And then that leads to circuit dysfunction and ultimately the symptoms over time. And there's a firm rationale for ketone metabolic therapy. There's already phase 1 clinical trials. There's 2 on clinicaltrials.gov. But from our research and other research, ketones are neuroprotective, cardioprotective, anti-inflammatory. They also have a protein muscle sparing effect. So that could be, and also traumatic brain injury is associated with post-traumatic epilepsy, psychiatric conditions. So that includes major depression, anxiety, and also substance abuse. And everything that I just mentioned there, that are 3 emerging applications for ketone metabolic therapy. Or seizures, it's already been around for over 100 years. But seizure disorders, psychiatric conditions, and substance abuse, if you go on clinicaltrials.gov, there's already clinical trials in process right now using exogenous ketones or ketogenic diets to treat those things. And with traumatic brain injury, I think it's just, there's a firm rationale for a metabolic intervention. And the framework, traumatic brain injury requires a very complex multidisciplinary care. But you have the acute phase, the subacute phase, and the recovery phase. And those 3 phases, which are characterized in a recent Lancet Neurology paper, those 3 phases, metabolism is critical for those 3 phases. So, brain oxygenation, but stabilizing brain metabolism is very critical for the acute, the subacute, and the recovery. And I think that's where ketogenic metabolic interventions, I think, could really be very important for that. And especially for the follow-up when TBI patients are getting psychiatric conditions and potentially post-traumatic seizures, it would be a really good idea to get them off the anticonvulsant drugs that they're often given and to give them ketone metabolic therapy, because many of these anticonvulsant drugs can impair your ability to recover from traumatic brain injury. So it just makes sense to do this. And I'm really, motivated and inspired to hopefully make some inroads from preclinical models to clinical research on this.
So following up on that, what population seems to be the most at risk for traumatic brain injuries? I don't mean occupationally, like the military or boxers or something. I mean, otherwise, what population demographic would seem to be most at risk and why?
Yeah, I mean, historically, it was sort of young kids and young athletes, and also, kids getting their license and driving cars and getting in accidents. I've had actually 2 of my friends from high school died of traumatic brain injury from car accidents. And so it was pretty traumatizing. But over the last decade, there's been over a 35% increase in older adults getting, it's actually become like a health crisis, a major financial burden. So you have, 2.8 million TBIs annually in the US and $76 billion economic burden for TBIs. So falls are really the leading cause of traumatic brain injury. And falls and also motor vehicle accidents. And an older adult that's over 75 has a 4 times greater risk of fatality if they do fall or if they get into an accident. And a relatively mild traumatic brain injury in an older person that a younger person can shrug off can be fatal. And that is a really important kind of consideration there. So yeah, I think the older population, for various reasons, mostly really because of frailty. So older individuals, not everyone, right? So you could have a 50-year-old that's frail and a 70-year-old that's in really good metabolic health and strong. And the 70-year-old or 75-year-old, the outcome will be much better just due to their metabolic fitness.
One of the things that when I asked the question, I didn't ask it very artfully. One can imagine, for example, young people having lots of TBIs because of the things young people do, right? It has nothing to do with their biology. It has to do with the insults that their brain endures. And older people, I think, have a double whammy. They fall much more and hit their heads quite a bit, but also their biology is different, right? They're much less resilient. I think everybody gets the first one, but it's probably important that the listeners understand the second one as well.
I can sum that up real quick. Yeah. So there's been a new characterization of traumatic brain injury in 2025, where the new characterization, it's called a practical framework to characterize acute traumatic brain injury. It looks at the clinical effect on the brain, the biomarkers, and also the imaging. But included in that is something called modifiers. And essentially, we know that the pre-injury patient characteristics are essentially the quote-unquote modifiers. So now when someone does come in to get evaluated for traumatic brain injury, they look at a number of different things, including age is one of them, but metabolic and vascular comorbidities, frailty, dementia, common medications, blood pressure medications, anti-psych drugs, and substance abuse, prescription drugs and also drugs of abuse, which could be alcohol or other things, increase the vulnerability of those patients to the injury and the recovery. And this is tightly coupled with the age of the individual, right? So aging is going to impact metabolic and vascular comorbidities, frailty, obviously dementia, and medications. Older people take more medications. So, and also if you've had a traumatic brain injury earlier in life, you are much more likely to get dementia too. So these are things that need to be tracked kind of over time. But now the new framework for evaluating traumatic brain injury is really focused on aging, and metabolic comorbidities are the highest sort of factor involved in someone's recovery from TBI. And this is finally being acknowledged.
That's really good. I'm happy to hear that.
So, Dom, this has been a lot of fun. So just a side note, several years ago, you and your wife, Chilla, bought some acreage in the countryside and started farming. And we know that farming is a lot of hard work. So are you still enjoying it?
Yeah. It's creative downtime for us, and we love being around animals. We got cows and sheep and our dogs, of course. Keeps us very busy. Very grateful to be in nature. It can be challenging, and we're always learning. After the Hurricane Milton, I'm actually still cleaning up, and this weekend I will be cutting up trees, and there's so many trees down from the hurricane even a year or more later. But we're really enjoying it, and thanks for asking. Yeah, we just love living on the farm.
That's awesome. And I had the chance to see Chilla actually back in the fall. I briefly got a chance to catch up with her at a diving show. So how is she doing?
Oh, yeah, she's doing great. She's in LA now, and she's really busy sort of advancing the science. She's publishing quite a bit, working on translating the science into the consumer, right? She's testing different formulations, bringing them to market. Still very involved in her marine biology research, studying manta rays, and she does that in Kona, Hawaii, where she looks at the various adaptations of mantas and also their cognitive function. So yeah, she's still very busy on the farm, but also busy publishing and also busy in her marine biology work.
Well, Dom, it's been so great to catch up with you. And again, congrats on all the fantastic work. And thank you for joining us.
Thanks for having me. Appreciate it.
Absolutely. It was wonderful to have you on.
Thank you.
STEM Talk.
STEM Talk.
Well, first and foremost, I have to say Dom is one of the nicest researchers and scientists that I've ever known and worked with. He's such a great guy. It's just been a pleasure to work with him I've known him for almost 2 decades now, which is crazy. He always has such a good attitude. And in today's interview, you could really tell how much he loves the research that he's doing. And over the next few years, we're going to be hearing a lot more from Dom's lab about ketone metabolic therapies as a complementary treatment for cancers, which is hugely impactful and really exciting.
Absolutely. The potential for ketone metabolic therapy as a way to help really manage devastating diseases like glioblastoma is particularly encouraging. With 49 authors working on the review paper in BMC Medicine that we discussed today, you can tell that this is a therapy with lots of promise and it generates lots of interest. Dom is at the forefront of this research, not only for glioblastoma, but also for other cancers and other metabolic disorders.
Absolutely. This is Dawn Carnegie signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.